just to show you why it's called duodenum. Duodenum is 12. And it's not 12 inches or 12 centimeters. It's basically 12 things. It's not actually like these, but I couldn't find any picture of it's 12 digits, right? So it's typically we have 10. So it's like this and add 2. So these are 2, but it's not that they're spread. So it's like this, 10, and then just add 2 fingers, right? That's the length of your duodenum. Okay, your 12 fingers is basically your duodenum. That's the length. So it's pretty short. What else do we have here? So bile is secreted from the liver, and pancreatic juice is secreted from the pancreas. And you'll know more than you want to about the components of all of these. And here at the end we have the hepatopancreatic ampulla. The ampulla is an expansion, a sac-like structure coming from that short duct. And then we have a sphincter. We're familiar with sphincters already, right? So that's the hepatopancreatic sphincter. Nothing to remember, just to think about how you'd call it, right? Hepato for liver, pancreatic for pancreas. Talk with each other. Tell me system B, very specific, as far as you can. Tell. Think about the logic. Maybe I want to be more specific in the questions, so not to mislead you. I'm not talking nervous and endocrine system. Of course, they're both involved. I'm asking nervous system, which parts of the nervous system? That's my question. Help digestion or prevent or inhibit digestion? And this is, I'm asking you this question not for the knowledge part, but for the thinking process part. This is a good example of something that you can easily get it wrong, just because of the not contracting and so on. Draw it, even if you don't draw it physically, draw that scenario in your brain, right? So if I'm telling the sphincter don't contract, it's going to be relaxed, right? So am I going to have more or less bile and pancreatic juices reaching, I should say, the small intestine? More. Is that going to help digestion or not? Yes. So is that going to be parasympathetic or sympathetic? Parasympathetic, because we promote digestion. So parasympathetic and enteric nervous system, when we need additional pancreatic juices and bile to continue digestion and absorption in the small intestine. Again, we're coming back, we need to think, we need to put ourselves in the small intestine and we'll ask, what do we need now? If I need some help, and we already learned that these are accessory, even if we did not talk about the details, these are accessory organs, we need some help. So you're going to call them and ask them, please help us, right? Okay. Any questions? Let's talk about the small intestine. Function follows form and what we would expect. Just like in the stomach, we had our third layer of muscle. Here we have our own anatomical features that will be adjustable and adjusted for the function of the small intestine. The small intestine needs to deal with a lot of kind, and it needs time. So it also needs a lot of surface area. So how do we create surface area? By folding, right? So the whole intestine, especially the small intestine, is fold within a fold within a fold. We have 3 different levels of folding. Okay, fold within a fold. The first one is what we call the circle of folds, right? So if you go, you can do it anytime you want, not now. If you put your hand inside your mouth, reaching all the way to the small intestine and just feel it, it will not be really smooth. It has these folds like you have here. Okay, so here's 1, 2, 3. Of course, we have many more than 3. Increasing the surface area, and it's kind of like, it's not just the surface area, you also slow the movement, right? It's like speed bumps. The car has to go slower. So this is how it looks like. These are folds and folds, and if you are a food or a digested food that is running right now, you need to go down and up and down and up and down and up. That means you're slowing down everything. And you have time in the small intestine to complete your digestion and to absorb everything that you want. Maybe not everything, but as much as possible. If we look at one of the circular folds, we can see the mucosa layer. We talked about mucosa and submucosa. We also have it here in the small intestine. you can see if we focus here that here even at a given circular fold we have these foldings of the mucosa. We call them villi in plural or villus in singular. Finger-like extensions of the mucosa. These are not cells, these are the whole mucosa. So there are many cells in each villus. So when we zoom in, this is how it looks like. Okay, here's the mucosa. You can see it's folded. Here are the different villi. Here's one villus, a second, a third, a fourth, and a fifth. And the fifth was cut so we can see everything in this mucosa over there. And the first thing that we see are lots of cells. So I hope you can see that. 1, 2, 3, 4, tons and tons of cells. There are different origins and different types, and we'll talk about the types. But the main cells here are the absorbing cells. Again, absorbing cells of the small intestine that we call enterocytes. Cardiocytes, melanocytes, and so on, these are the enterocytes. They are the cells that are responsible for absorption of molecules from this lumen, the lumen is here, into the blood and the lymph. Any questions about the anatomy so far? To make sure we're not missing. Yes?
Does the bumpy texture help with mechanical digestion too?
Does the bumpy texture
Help with mechanical digestion at all?
helps with mechanical digestion? Mechanical digestion at this point was basically completed with the stomach. So right now what you have is only fluids. It's fluids only, or almost only. So mechanical is not a big deal here. Probably maybe some small contribution, but minor if any. Okay? We're all set and oriented with the villi and so on? Okay. So these are finger-like extension. Now this is zooming in into the enterocyte. So here's one enterocyte and the next one next to it and another one next to it. Here's the nucleus of the cell and all the organelles, just a regular cell. When we look at the membrane of this cell, what else do we see? Even that is folded, right? These are called the microvilli. So that's folding at the membrane level of each of the enterocytes. And this one carries, again, increases the surface area even more, but it's beyond that. Because those membranes, these are the apical membranes, the membrane facing the lumen. These are the basal and lateral membranes, side domains of the membrane. This is the apical membrane, the one facing the lumen. They're not just empty with some receptors like all cells they have a lot of enzymes sitting there, and they're called the brush border enzymes. They're called the brush border because they do look like a brush because so many of them. And this is the electron micrograph, sorry, and these are the microvilli. All of this is just one cell. So these are the microvilli, 1, 2, 3, and 4. And all of this right here are all these brush border enzymes. You do not need to know the names of the brush border enzymes specifically. Just think about them as a group. And the job, they assist and are essential in the digestion and in the absorption, but we'll just talk about digestion at this point, of carbohydrates and proteins in the small intestine. Next week, when we'll talk about the different groups of macromolecules, we will see how important they are. Fold within a fold within a fold. Circular folds, villi, microvilli. Questions? Let's talk about what all these guys secrete. So we had in the stomach gastric juice, in the intestine we have intestinal juice. We zoom out now, and we look on these cells. We look at the histology. So as I said, most of these cells are the absorptive cells. They are what we call the enterocytes. We also have goblet cells. Goblet, an old wine glass type of glass. This is basically the reason they just look like that. Goblet cells are found not just in the intestine, but in many different organs. And they will be the ones that secrete the mucin, that will become a mucus. We also have enteroendocrine cells, just like we had in the stomach. In this case, it's not gastrin. Gastrin is a gastric hormone. In this case, we're talking about the intestinal hormones. One of them we just mentioned, secretin. CCK will be another one. These are not the only two. I'm just going to mention these two. As far as I'm concerned, for the sake of this class, you need to remember these two. That's it. But just know that there are more. Okay. Well, we're going to do the note card thing. Yeah, we'll do the note card thing right now. What I want you to do to look at is to think about the enterocytes now, and to think about the function. Again, we have food that is not food anymore. It's digested food, it's chyme, or partially digested food, I should say. And we are going to continue to digest it, but then we need to absorb, because if the food is just moving and going into the anus, nothing happened, right? We need to absorb these. Where are we going to absorb it? Through the enterocytes, and eventually into those capillaries that you see here, and from them, we'll talk about later on. But that's the idea of absorption. So based on that, and based on other things that we learned in the class, I want you to answer 2 questions on a note card, okay? Please try to do it in groups 2 or 3, but not just with yourself. I really want you guys to think together. The first thing I want to ask you, which type of capillaries would you expect for this specific function? We learned about capillaries in the previous section, so remind yourself, okay? Or if you don't remember, go back to your slides. But the second question is more important. Based on what you answer in the first, on what type of capillaries you would expect here, what does this tell you about the physical interaction between one enterocyte and the other one? Is it going to be very tight, not so tight, completely permeable, half permeable? Think about what you would expect based on the expected functions of this whole system. Okay? I'm going to give you 2 or 3 minutes to think about this. And at the end, what I want you to do, to bring it all to the right, all the note cards, and all the way to the bottom for Brian. Brian will give it to Dana, then I will give it to you. You remind me. Fenestrated. Majority. BBB type. Okay, so most of you said fenestrated. Why fenestrated? What's the logic? Why would you be a fenestrated capillary if you're sitting there? You are correct, by the way. Yes?
So essentially, he doesn't break it up. I think these are like subcategories.
You want to maximize
So it's either
absorption.
continuous or
We are now
frustrated
dealing
in this case,
with
or BBB, which is really
tons
tight.
of micromolecules, amino acids,
fenestrated
simple
capillary is what
carbohydrates,
goes
simple lipids that were just absorbed after all this work, right, from the lumen. And we need to bring all of this stuff into the liver and eventually to the circulation. That's a lot. It's like if we just produce something, right, we bring it to the train. Let's imagine we need to, the propagation will be through a train, okay? Then I cannot have this train to be too selective. I cannot have a conductor that will say, wait a second, show me this and show me that. I have all my produce, let's just put it on. So I'm going to use fenestrated. Not only that they're not so tightly sealed between one cell and the other in the capillaries, between one endothelial cell, they're going to have holes. So I can take all the stuff, all the products, the amino acids, and throw them through the windows into the train. Just go and deliver. Right? That's the idea. But can be a potential ramification in what do we need to do? What does it tell us about the interactions between the enterocytes? It has to do with something we already discussed in class. I just want to make sure we understand this. This is a critical point. Miranda. It says something with tight junctions. What would you expect? More or less the normal tight junctions between the enterocytes? Less or more? Less or more? Most of you say less. Anyone says more? Ryan was about to say more or less? Okay. Oh, wait.
Maybe more so it keeps the other stuff.
Here is I'm in the lumen, okay? I'm now an amino acid right here in the lumen. Here are the cells. Tight junctions, more or less, right? I know that once I pass this, here's the train, right? I'm sending everything through the windows, the fenestrated through the capillaries, right? So I'm very efficient here, but I'm not very I'm not selective, right? Like everything I can, not everything of course, but everything that I can, I'm just pushing it. So what could have happened if I let it go here and if I have less tight junctions? What might possibly go through? Pathogens, bad stuff, things that I don't want to. Remember, Our internal system, it's an internal system, but it's open to the outside. Yes, I try to kill it in the stomach, and many pathogens will be killed, but there's a lot of bad stuff still here. So I have to be extremely selective at one point. And the selection will be in an extremely tight junctions, heavily concentrated tight junctions between one enterocyte and its neighbor. It's not going to be BBB style, not at that level, but it's going to be very tight. Now, it doesn't apply to all the time in our life. For example, when we are very young infants, it's not. But in general, that's a problem. And that problem, if it's not, when I say problem, I mean when it's not tight enough, it's a problem that many of us actually encounter. And one of the factors that lead to intestinal-related chronic issues. Okay? Many of you heard about celiac disease, right? Celiac sensitivity to gluten. It's composed of different factors. And the most important one is our own immune system is attacking a specific peptide, and therefore all that region. But if I'm that peptide that is part of the gluten, the protein, then I don't even need, I cannot even penetrate, right? I cannot even reach the immune system. But the problem is that in celiac, basically, there's a lesion here. And this affects the tight junctions and I'm penetrating. So you have a hole, if you will. Sometimes it's called the leaky gut. It's not the scientific name, but we see some pathologies that have to do with what we call sometimes the leaky gut. A break, a lesion inside in those selective border of the intestinal cells, and as a result, no selection, a lot of stuff goes in, but then a mess.
So the enterocytes.
Okay, I want to continue to make sure we cover the next, just the next part. So just to summarize this.
Outer layer cells, enterocytes. And
All right,
then once they
do
get
it.
through the cells, they just push them onto the train. They push them into the blood as quick as possible. And then that's selective.
Okay, so the
Okay,
canal
just do
is
it
like
this way.
on the outside with the enterocytes.
Yeah, the enterocytes are
Okay,
the cells
fenestrated
they're
capillaries
called enterocytes, right?
to allow
Like
an
in-
efficient loading
they're
of
just the cells that
nutrients
line it,
onto
those
the
red cells.
capillaries. And
Those
we
guys
should
are selective
expect
and they have
tightly
tight
sealed
junctions.
junctions between the cells.
Between those cells. Between the absorptive cells, pretty much.
We also see another
Then
thing,
once
and
they
that's
get
the
absorbed,
green big
they've already
thingy. What is
passed
that?
that checkpoint, then it doesn't matter.
That's the lymphatic system.
Like if
That's
the
where we come
capillaries are
back to
tight
the
because
absorption
they've
of
already
fats. We call
been
this the
screened.
lacteal, that specific
It's
lymphatic
like you only have to go
vessel.
through one security checkpoint to
It's
get in the airport.
wider,
So
so it's
you go
bigger
through
than the
the TSA
capillaries.
check.
As we will learn
And
next
that lets you
week,
into the airport,
lipids
but then when you
undergo
actually board the
packaging
plane, you
because
don't
they're
have to get
hydrophobic,
checked.
so we cannot have them just run in the
Okay.
blood. They're not
Same
soluble. So when
already
you
been
package
checked
them, the
once.
problem is the package is just too big for the capillaries. So instead we have the lymphatic system carries them eventually into the vein. Remember, the lymphatic system is not independent of the blood. It is drained into the veins in two spots. And this is how we're going to have lipids joining the circulation. No, it doesn't want to respond. Okay.
So think of it like that airport. You have to go through one checkpoint to get into the airport, but you can go to a restaurant after that, you can walk around. And once you actually get on
We
the
talked
plane,
about the
there's no
villi, right? Those ridges, if you will.
Exactly.
But we also have crypts that look not very different from the glands that we saw right in the intestine. In the crypts, what we find are the cells that are going to secrete the intestinal juices, but the intestinal juices are mainly a mixture of water and electrolytes, not too much. I do want to mention the fact that they are providing us with an alkaline solution because of this type of electrolytes. Why do we need an alkaline solution being secreted from the intestine? Because we need to neutralize, remember, the acidity that comes from the stomach. Here's your very first source for alkaline solutions. We're going to talk about three important sources, and we have to have it. In the crypt, we also find the stem cells that are going to divide endlessly with no limit because they always need to provide a new series of enterocytes. The enterocyte that you see right here, the one that I'm showing you right here, was born in the stem cell, was being pushed and pushed and pushed and pushed until it made it here. It's going to die in 4, 5, maybe 6 days. That's it. It's going to face an extremely harsh environment physically. and it's going to die. And we need all the time to supply more and more cells. You can actually see, I'm showing you again that circular folds image. It's hard to see the orange ones. Orange one is a label, a marker for cell death. These are the ones that just died, the cells that are shed off. It's really annoying that it doesn't work by itself. Okay. Okay, I'm just going to say we have 2 minutes. I'm just going to briefly talk about the liver. The main thing you need to know about the liver is what you read. The intestinal cells secrete a mixture of water, as I said, alkaline juice, but that's not sufficient for all the digestion and absorption. So we need to get some assistance, and the assistance comes from 3 different sources: the liver, gallbladder that assists the liver, it's actually storing, it's not making anything, and the pancreas. So in the 2 minutes that are left, I just want to talk briefly about the liver. And again, it's mainly going to be through your readings. The liver is making bile. The hepatocytes, the basic cells of the liver, are the ones synthesizing the bile. What is bile? Bile is a mixture of molecules that are, first of all, is extremely alkaline. And we just talked about why we need it to be alkaline, right? To neutralize the acidity. Bile is everything you can think of regarding lipids. It's mainly composed of bile salts. Bile salts all started from cholesterol. We talked last time a bit about cholesterol. So they're all derivatives of cholesterol, and what they do, they emulsify the fat. Lipids don't like water, right? So they form these big droplets of fat, right? It's not these big, but I'm just showing it like this, right? Running in the juice. We have enzymes that will digest fat, but it's hard to access those lipids inside the big droplet. So what we do, we use soap, a detergent. Bile salts, they are just like soap. They are breaking the big droplet into smaller and smaller droplets. This is what soap does. They what we call emulsify it. So they will be accessible to enzymatic digestion. Other things we'll find there, I know we're about to be done, now we need to leave. Bile pigments, cholesterol itself, the neutral fats, phospholipids, and waste, everything can happen. Okay? We're not going to continue. We're going to continue next week, right? Today's Thursday. Great weekend.